So the next part of this lecture is really going to be discipline specific. So we're going to spend quite a bit of time talking about plant science research and some different things to consider when setting up a project there. And we're going to spend less time at the end talking about animal projects and what to consider setting up there. And the reason for this, you're going to totally notice an unbalance between plant information and animal information. But plants, for the most part, don't move, right? And yes, they are alive, but they are usually non-moving plants. Animals are a little bit harder to deal with, and there's so many individual considerations that have to be made for individual research projects. And so I'm just going to give you a few of the basics on the animal side, but I'm going to give you a lot more detail on the plant side. All right, so let's dive into the plant side real quick. So in plant experiments, you really have to consider the area that you're conducting the research in. So are you using just demonstration plots, or are you actually doing a replicated field trial? And the reason that this question comes up is because it all goes back to your audience. So if you're an extension agent, you may not need to publish in a peer-reviewed journal, but you may need to, have some information about, rice variety or alfalfa variety for the clientele in your county or your region. And so a demonstration plot can absolutely give you that information without doing a full-blown study. However, if you are a researcher, you definitely, and you're wanting to get published in a peer-reviewed journal, you definitely need to make sure that you're in a replication type scenario. And so this goes back to the importance of replications. And again, this is a little bit of a reminder from the principles of experimental design. But replications in plant science research is really important. You need to make sure that you have adequate experimental units assigned per treatment to make sure that you can detect a difference if there's one there to be detected. So some common experimental designs that plant science studies will use is a completely randomized design. That's the simplest. We're going to learn that inside and out. If you have a known source of variation, you could use a randomized complete block design, which is an RCBD. And then plant experiments, if you're trying to look at multiple tractors or multiple sets of treatments, they will actually use a split plot. Example, and I'll give you an overview of what those look like here in a few minutes. And so some other considerations that researchers need to think about is, are their plots small and going to take up a minimal amount of land, or are they large plots that are going to take up a large amount of land? So for example, if you have small plots that are like 1 by 1 meter squared, you can afford to have, most likely, multiple replications of those treatments. However, if your plots are 30 by 30, that is a lot bigger, and so having more replications sometimes isn't feasible. So you have to make sure you have a good handle on what land you're using for your experiment. And that's important for a lot of reasons, but plot maintenance is the big one. So if the plots take a lot to stay maintained, like you have to make sure that border rows are staying clipped and applying treatments on a regular basis, you may want to do that on a 1 by 1 meter plot versus a 30 foot by 30 foot plot. All right, so with the objective of a plant experiment, keep in mind that if you are like an extension agent and you are just generating information for growers that's not going to be peer reviewed, you can actually use really simple qualitative comparisons. So qualitative, keep in mind, are categories like yes or no, pregnant, not pregnant on the animal side, yes it grew, no it didn't on the plant side, those type of things. And so some advantages to these qualitative comparisons are that they're really simple to coordinate and assess. And you can do that in a pretty timely fashion. Some of the disadvantages are it's really hard to extrapolate qualitative data to into a quantitative study, especially if you want to later on get it published in a peer-reviewed journal. And so qualitative studies may oversimplify the issue and not really be representative of what is happening in the greater population. So let's look at some examples of of these designs. So if we had different rates of nitrogen that we were trying to apply on a field crop, we might be able to apply them to a field in this type of orientation, to where this part of the field is going to have the high nitrogen treatment, this part of the field is going to have the low or the normal one, and this part is going to have the low nitrogen treatment. Okay, so that is a pretty straightforward example, but in subsequent slides I'm going to show you it can actually get a little bit more complicated the more things that you add to it. Alright, so when we think about a replicated field plot, so using that information for like a peer-reviewed journal, the objectives here have to change. You have to create objectives that are going to statistically compare treatments. So it could be whatever your treatments are, varieties, etc. And so some advantages to a replicated field plot trial is that the results are recognized by the scientific community, usually, and they're suitable for making really big decisions, like production-based decisions. Some disadvantages are they're way more complicated to conduct. And so when we look at a replicated trial, the way that we orient our experimental design gets a little bit more complicated. So again, if we were using that same scenario where we were trying to determine the rate of nitrogen on a field crop, and we had high, normal, and low, in order for it to be a replicated trial, we would have to actually have 3 different fields in this trial. And so those fields would then become blocks, and within each field would be at least one of the treatments. And so big differences between like a demonstration plot in a qualitative study versus a replicated trial in a quantitative study. So researchers are always plagued by the question, how many replications are enough? And so generally the consensus is the more the better, right? So it increases degrees of freedom, it's a better representation of a population, it's easier to differentiate at similar means, but that mentality can get really tedious really quickly because you have to consider how much land you have, how much time you have to collect the data, and how much material you have. So it can be really costly if you get way too many replications over and above what you need. So this is why we do statistical power calculations. Okay, so here's some general rules of thumb when it comes to plant science research. The minimum number is going to vary depending on the discipline and the researcher. I'll just say that up front. That's my disclosure. But the absolute rock bottom minimum number of replications that you can have in plant research is 3. Okay, if you are doing a fungicide trial, then peer-reviewed journal articles like to see at minimum 4 replications. And if you're doing something associated with soil, like trying to detect a soil-borne disease, which includes nematodes, then peer-reviewed journals like to see 5 and even 6 replications. And that's because all of those things are highly, highly variable. And so when it comes to like the soil example, the organisms in the soil are really unevenly distributed. So you have to have more replications to try to capture what is really happening in the population. So when it comes to soil variability in plant experiments, there are some things that you need to take into consideration. If you have a field and you're doing a field trial and you have really random soil variability, your plots have to be larger to capture that variation. However, if you have a field that is on a gradient, like let's say that it's positioned next to the Sacramento River, on the non-river side of the field, you may have, really decent clay-based soils. However, as you move closer to the river, that that clay soil may change into more of a gravelly soil. And so you have that gradient that can really affect how the treatments perform in those different treatment groups. And so in that case, you want to make sure that you have a rectangular plot to capture the whole entire gradient. And if you have an area that is where the soil is unknown, first of all, that's not really an excuse anymore. We have this wonderful tool. It's called Web Soil Survey, and it will give you, at the bare minimum, the soil variability in an area. But if you have an unknown variability, a square plot is always going to work best. If you are looking at a cropping system, to where you're harvesting crops and considering tonnage and yield and that type of stuff, the minimum size of the harvest plot needed is really a big consideration. So, for example, if we were growing lettuce, you might be able to get a good idea of lettuce yield with 10 plants. Okay, but with rice, you probably need 5 meters squared to detect a difference if there's one there. And with corn, which is even more variable than rice, you actually need 10 meters squared. All right, so when it comes to small plot research projects, it's really, really important that the researcher and the project stays in the research area stays very neat and very precise. So the smaller the plot, the more neat and tidy it needs to be. So small areas actually magnify issues with yields and then we try to convert them to a larger basis. And so, for example, if one small plot got overwatered because it was closer to, the source of the water or something like that, it wouldn't matter what treatment it would be, it would tend to have probably better yields. And so then we would take that into consideration when trying to apply it to tonnage on a bigger basis. And it wouldn't necessarily be because of the treatment, it would be because it was in a favorable location. So those are all things to take into consideration.